Good day everyone, I am Jan Cleo Caranog and today I'll be discussing the molecular photoluminescence spectroscopy and its subset, molecular fluorescence spectroscopy. So photoluminescence spectroscopy is a technique used to study the materials by analyzing the light they emit after absorbing energy. When a material is exposed to the light, its electron get excited and move to a higher energy state. As these excited electrons return to their normal state or ground state, they release light. By studying this emitted light, scientists can understand the material's electronic structure, detect any impurities, and evaluate its overall quality, especially useful in studying semiconductors. So the specific type of this technique is called the fluorescence spectroscopy. This focuses on the immediate emission of light after a molecule absorbs it. When the molecule absorbs light, one of its electrons jumps into a higher energy level. It then quickly drops back to its original state or the level, releasing the light in the process. What makes this technique powerful is its high sensitivity and accuracy. It's commonly used to detect and measure very small amounts of substances in complex biological or chemical samples. So at the end of this lesson, you should be able to define photoluminescence spectroscopy, determine the form of photoluminescence, process of photoluminescence spectroscopy, and learn about the relation between absorption and emission spectra. So first, we have the forms of photoluminescence. First is the resonant radiation, second is fluorescence, and the third one is the phosphorescence. So the first one is resonant radiation. It is a process with a material observes a photon of a specific wavelength and almost immediately emits a photon of the same energy or wavelength. And what makes this process unique is that there are no significant changes or transition in the internal energy of the materials between the absorption and emission stages. So ibig sabihin, hindi nagbabago ang chemical structure nito. Kung ano yung energy na na-absorb, yun din yung nire-release nito. This entire process happens very quickly, typically within 10 nanoseconds, which means it's an almost instantaneous reaction. The second one is fluorescence. It is a phenomenon where a chemical substance absorbs light and gets excited to a higher energy level. But unlike resonant radiation, it doesn't immediately release the energy. Instead, the substance first undergoes internal energy transitions, meaning some of the energy is used or lost inside the molecule before it returns to its ground state. And because of this, the light that is emitted has a lower energy compared to the light that was originally absorbed. That's why the color of the emitted light is usually different. This process happens very fast, but not instantly. The lifetime of fluorescence usually ranges from 10 minus of 8 to 10 minus of 4 seconds, which is still very quick, but it allows time for internal changes before the light is emitted. The third one is phosphorescence. It is also a type of radiational transition where a substance absorbs light and stores energy. But unlike fluorescence, the absorbed energy in the phosphorescence undergoes intersystem crossing, which means the energy moves into a state with a different spin multiplicity, usually from a singlet state to a triplet state. Because of this extra step, the molecule holds on to the energy longer before releasing it as light. Kaya naman, the lifetime of phosphorescence is much longer, usually between 10 to the minus 4 to 10 to the minus of 2 seconds. That's significantly slower compared to the fluorescence, which only lasts up to 10 minus of 4 seconds at most. So since the molecule stays longer in the triplet state, there is also a bigger chance that it will return to the ground state without emitting light, making the phosphorescence rarer than fluorescence. So let's come now to the instrumentation used to study this phenomena. And to measure photoluminescence, we use a device called a spectrofluorometer. This is an analytical instrument designed to detect and measure fluorescence emitted by a sample. It works by scanning either the excitation wavelength, the emission wavelength, or sometimes both. depending on the type of analysis needed. This instrumentation is highly versatile. With the help of additional attachment, it can also study how the fluorescence signal changes over time, temperature, concentration, or other environmental variables. This makes it useful in wide range of scientific and industrial application. So in this diagram, it illustrates the components and their arrangement in a spectrophlorimeter, also known as the fluorescent spectrophotometer. In most spectrophlorimeters, the light source commonly used is a 150 watt ozone-free xenon arc lamp. This powerful lamp produces a broad spectrum of light, making it ideal for exciting various types of sample. The light coming from the xenon lamp is focused by an elliptical mirror into excitation monochromator. It passes through a quartz window which serves a two important function. First, it vents in the heat produced by the lamp, preventing the damage to the system. And second is it provides safety, ensuring that the intense light doesn't harm the user or equipment. This setup is carefully designed to achieve full spectrum resolution, meaning it can cover a wide range of wavelengths with a high precision. It also minimizes optical problems like spherical aberration and redifraction, ensuring accurate and clear result. So the monochromator is a key component in spectrophlorimeter. It's responsible for selecting specific wavelengths of light, either for exciting the sample or analyzing the emitted light. So there are two types of monochromator used in the system. First is the excitation monochromator. This selects the wavelength of the light that will excite the sample. Second is the emission monochromator. This selects the wavelength of the emitted light that will be measured. So this monochromators is used entirely reflective optics, which helps maintain high resolution across the full range of the spectrum. Reflective optics are used instead of lenses to reduce spherical aberration and redifraction, and which can distort the light and the effect and accuracy of the result. So inside the monochromator, the most crucial part is the reflection grating. This component is responsible for dispersing the incoming light into its component wavelengths. It does this by using the tiny grooves that are aligned vertically on the grating surface. These grooves interact with the light and separates it based on the wavelength. To scan or to analyze the spectrum, the grating rotates, allowing the different wavelengths to be selected. In most systems, the grating uses 1200 of grooves per millimeter, which provides high resolution. They are blazed, so meaning it is optimized. For two specific wavelengths, they have 330 nm for excitation and 500 nm for emission. Since gratings are sensitive and can oxidize over time, they are coated with protective layer of magnesium fluoride. This layer helps prevent damage and ensures long-term performance and accuracy. So another inside of the monochromator, we have the flexible slits located at both entrance and exit. These slits control the bandpass of the light, meaning how much light and which range of wavelengths are allowed to pass through. Wider slits allows the light to pass through, which can increase the fluorescent signal. However, this comes at cost of less resolution. You get more light but less detail. Narrower slits, on the other hand, give you a higher resolution and better detail on the data, but they also reduce the light intensity, which makes the signal weaker. So there are two main shutters used in the system. First is the excitation shutter, located just below the exit slit of the excitation monochromator. Its main purpose is to protect the sample from too much light exposure, which can cause the photo bleaching or photo degradation. In short, it prevents the sample from being damaged by continuous light. Second is the emission shutter. It is placed just before the entrance of the emission monochromator. Its role is to protect the detector from being exposed to excessively bright light, which could affect the accuracy of the result or even damage the detector. So next is the sample compartment. It is where the actual sample is placed for analysis. It can include optional attachment and fiber optic bundles to make the setup more flexible and adaptable to different types of experiment. The fiber optics serve two main functions. First is they deliver the excitation beam from the light source directly to a remote sample, especially useful if the sample is not placed directly in the instrument. Second is they also collect the emitted light, the fluorescent or the phosphorescent, and they return it to emission monochromator for analysis. So next is they use two detectors, the signal and the reference detector. First is the signal detector. This is responsible for detecting the fluorescent or the emission coming from the sample. It uses an R928P photomultiplier tube, which is very sensitive and ideal for photon counting. In short, it counts the number of the light particles or photon emitted by the sample. Second is the reference detector. This uses the UV enhanced silicon photodiode, and its job is to monitor the output of the xenon lamp. This helps the system make correction over time and wavelengths, ensuring that any changes in the lamp intensity don't affect the accuracy of the result. So let's come now to the relationship between the absorption and the emission spectra. When a molecule absorbs light, it gets excited from the ground state to a higher energy level. Before it emits the light, it releases some energy inside, then goes back down by emitting light, and this is called the fluorescent or the phosphorescent. Because some energy is lost during the process, the light that comes out or the emission has a lower energy than the light that was absorbed. That means emission happens at a longer wavelength than absorption. If the energy level transition are similar, the shape of the absorption and emission spectra can look like mirror images of each other. Also, after absorbing light, the shape of the molecule changes a bit. When it emits light, it returns to the ground state, but it keeps the shape of the excited state, and this also causes the emission to have a less energy. Now, to better understand how forms of photoluminescence process work, we will also look at Jablonski's diagram. First is the diagram shows the different energy levels of a molecule. The ground state is called the SO. It's where the molecule normally stays. The excited singlet state is called the S1. It is where the molecule goes when it absorbs light. There is also a triplet state called the T1, which involves in the phosphorescence. Now, when a molecule absorbs a photon, it jumps from a ground state to the excited state. This step is called the absorption. After that, the molecule releases some internal energy, or so-called the vibrational relaxation, and then returns to the ground state, releasing light in the process. This quick emission of the light is called the fluorescent and happens within nanoseconds. Sometimes, the molecule doesn't go back directly to the ground state. Instead, it moves to the triplet state, which is a state with a different spin. From there, it is slowly returned to a ground state and emits light. This slower process is called the phosphorescence and can last from milliseconds to even seconds. So this graph is the absorption and fluorescent spectra of atrazine, a polycyclic aromatic hydrocarbon. On the x-axis, we have the wavelength in nanometers, which represents the different colors or energy of the light. On the y-axis, we see either absorbance or fluorescence intensity, which tells us how much of the light atrazine absorbs or emits at each wavelength. So there are two curves in the graph. The black curve represents the absorption of the spectrum of the atrazine. This shows the specific wavelengths where atrazine absorbs light and transition to a higher energy state. You'll notice several peaks, each corresponding to the light being absorbed at particular energies. These peaks are unique and helps us identify the atrazine. And the green curve is the fluorescent spectrum, which shows the light emitted by the atrazine after it absorbs energy and returns to its ground state. These peaks are shifted toward a longer wavelength. This shift is known as the stroke shift, and it happens because some energy is lost during internal processes like vibrational relaxation before light is emitted. And the green curve is the fluorescent spectrum, which shows the light emitted by the artesine after it absorbs energy and returns to its ground state. These peaks are shifted toward a longer wavelength. This shift is known as the stroke shift, and it happens because some energy is lost during internal processes like vibrational relaxation before light is emitted. Now, if you look closely, there are some overlap between the absorption and the emission spectra near the wavelength, which is the excitation wavelength used in the experiment. The structure of the artesine shown in the inset plays a key role here. It's altering the single and the double bonds allow it to interact with the light and the UV visible range, enabling both absorption and the fluorescence.